Okay, the part that refers to the organization of a skeletal muscle, it's very important. Let me go back to right here. Okay, so organization of a skeletal muscle is actually quite important. And what this is telling us is how skeletal muscle cells are organized within a muscle. So first of all, skeletal muscle cells are going to be bundled together by a connective tissue membrane called the endomysium. Endomysium bundles several of these. I'm sorry. Okay, let me go back. Skeletal muscle cells, each individual cell is covered by a connective tissue membrane called the endomysium. So it's like a sleeve that covers individual cells. Then several cells are going to be bundled together into fascicles by another membrane called the perimysium, and several fascicles will be bundled together by epimysium. So let me go to the picture here, and this picture is showing you an entire muscle. Okay, so it's looking at the entire muscle, like the biceps right here, and this is the entire muscle. So the membrane that covers the muscle on the outside is the epimysium. Epi means upon, and my means muscle. So there's going to be a lot of terms with the my root in the term, because that means muscle. All right, so the epimysium covers the entire muscle. Muscle cells are bundled into groups called fascicles. So this right here that has been pulled out is a fascicle, and the connective tissue membrane that bundles muscle cells into fascicles is called the perimysium. So here on the outside will be the perimysium. The other circles you see in the muscle are other fascicles bundled by perimysium. Then from the fascicle, one little muscle cell is being pulled out, and the individual muscle cell is covered on the outside with a connective tissue sheath called the endomysium. So this depiction here is a cross-section of the entire muscle. The circles, the large circles here, are going to be fascicles, and the connective tissue that is bundling muscle cells into fascicles is the perimysium. The connective tissue outside of the muscle, bundling many fascicles together, is the epimysium. Within a fascicle, the smaller red circles that you see there, those will be muscle cells. So this right here is an individual fascicle. So it's one of these circles pulled out, and the cross-section of the circle is what we see here. This is a muscle fascicle. So the connective tissue membrane surrounding the muscle fascicle is the perimysium. Now the circles inside are going to be muscle cells, which are also called muscle fibers. And the smaller circles within the muscle cells are going to be myofibrils or contractile proteins. Now we're going to pull one of these muscle cells out, look at a cross-section, and this is an individual cell, a muscle cell, which is called a muscle fiber. So the connective tissue on the outside of the muscle fiber is going to be the endomysium. On the inside, the muscle fiber is going to have many contractile proteins called myofibrils. Okay, so let's go at another depiction here. This is actually a picture of fascicles. So this will be one fascicle right here, and here's another fascicle, and you can see this entire picture is fascicles, which means that the connective tissue membrane right there surrounding the fascicle is the perimysium. The circles inside the fascicle are going to be the muscle cells or muscle fibers, and each individual muscle cell is surrounded by an endomysium. So we're going to have in the next picture a close-up of a muscle cell surrounded by its endomysium. So go back and review the organization of muscle fibers within a muscle. That is important for the test. Now, skeletal muscle fibers or skeletal muscle cells are highly specialized cells. These cells are specialized for contraction. They're called skeletal muscle fibers or just muscle fibers, but they're not talking about, the name does not describe a protein fiber, it's describing a muscle cell. Another term that we use for muscle cell is myocytes. So muscle cells are either called myocytes or muscle fibers. So like I mentioned before, the cell itself is very much specialized for contraction. It is a huge cell. Here you only see a portion of the cell. It is multinucleated. It has many nuclei because the cell is so big that it actually came about by the fusion of many cells together. And that's why it's multinucleated, because each cell provided one nucleus. Another characteristic of this cell is that it is filled with contractile proteins called myofibrils. So these contractile rod-like proteins called myofibrils are unique to these skeletal muscle fibers. The cell membrane of the muscle fiber, which will be deep to the endomysium, is called sarcolemma. So the sarcolemma is the cell membrane. And then superficial to the sarcolemma, there will be the endomysium on the outside. Okay. Now, covering each individual myofibril or contractile rod-like proteins is going to be this structure right here in blue, which is the sarcolemma. Sarcolemma, is the sarcoplasm. Is the sarcoplasma reticulum here, which is the endoplasmic reticulum of the cell. But in the muscle cell, instead of calling it endoplasmic reticulum, it is called sarcoplasmic reticulum. Also notice these yellow structures in the middle of the sarcoplasmic reticulum, right here and right here. These structures came about by the infolding of the cell membrane or sarcolemma, creating these tube-like structures called the T-tubules. So this is very unique to skeletal muscle. So that if we look again at these T-tubules that have sarcolemmas or sarcoplasmic reticulum flanked on either side, this is what we call a triad. So a triad is again unique to skeletal muscle. Only skeletal muscles have triads. A smooth muscle doesn't have triads. Cardiac muscle doesn't have triads. Triads consist of the infolding of the sarcolemma, the cell membrane, into a tube called a T-tubule. Flanking either side of the T-tubule is a layer of these sarcoplasmic reticulum, which creates these sac-like structures called cisternae. So a triad consists of two cisternae flanking a T-tubule, and the T-tubule is made from the cell membrane, from the sarcolemma. Notice the little red dots inside the cisternae. Those are calcium ions. So the job of the cisternae is to store calcium ions inside to be released when muscle contraction is needed. So you should definitely know the structure of a triad and the function of a triad. Okay. So these long rod-shaped contractile proteins are called myofibrils. Remember that surrounding the myofibril is going to be the sarcoplasmic reticulum on either side of a T-tubule, and that constituted a triad. right there. Okay. Myofibrils, in turn, are made of proteins, what we call thick and thin protein filaments, which are arranged in a regular pattern called sarcomeres. Sarcomere is called the contractile unit of skeletal muscle. Okay. So let's go back to the organization of skeletal muscles. First, we have the entire muscle, and the entire muscle is surrounded by a connective tissue membrane called the epimysium. The epimysium bundles together several fascicles. So now we're going to pull one of these fascicles and take a look at it. Fascicles bundle individual muscle cells. So each one of these larger circles inside this fascicle is a muscle cell. It is bundled together. These muscle cells are bundled together in the fascicle by a connective tissue membrane called the perimysium. Then we take a look at one individual muscle cell within a fascicle right here. And notice that we have a connective tissue membrane covering the muscle cell called the endomysium. And it is a sleeve that goes over the sarcolemma or endoplasmic reticulum. Remember, the muscle fiber or myocyte is a rod-shaped cell, and inside, those are the red dots you see here, it is filled with a contractile protein called myofibril. Myofibrils are surrounded by triads. Here's one triad, here's another one. And if we take a look at the myofibril, turns out it is made of repeated subunits of a contractile structure called the sarcomere. So the sarcomere is as small as we can go when we're trying to understand contraction. This is what we call the unit of contraction. Take a look at this picture again. This is the myofibril, and if you look, here's one sarcomere. Okay, here's one sarcomere, and then there's another sarcomere, and then another. So sarcomeres are the contractile units that make up the myofibrils. Okay, so let's talk a little bit more about the sarcomere, and you definitely have to understand what the sarcomere is all about. So the sarcomere, you definitely need to know these, is the contractile unit of the muscle fiber. So if you're trying to understand muscle contraction, you must understand how does the sarcomere contract. Okay, and you should definitely know and be prepared to identify the parts of a sarcomere. So first of all, on either side of these arrangement of protein fibers, of proteins, the sarcomere is flanked by a Z disc on either side. So here's a Z disc right there, which means this whole thing right here is a sarcomere. Then there's another one coming in here, then there's another one after that. Okay. So sarcomeres are flanked on either side by Z discs. Then they have two types of filaments on the inside. They have a thin filament right there, flanking a middle thick filament in the middle. Okay. Notice that the thick filament starts and ends right there. Okay. And the start of the thick filament all the way to the end of the Z filament is what's called the A band. The A band consists of an area where thick and thin filament overlap. Thick, thin, thick, thin, thick, thin. This is called the zone of overlap. So this is zones of overlap. So this is one zone of overlap right here. And here's another zone of overlap on the other side. Thin filament, thick filament, thin filament, thick filament, thin filament, thick filament, thin filament. Okay? In the middle of the zone of overlap, there is an area where all we see is thick filament. There is no thin filament in that middle area. This is what we call the H zone. Okay, and the line of proteins in the middle is the M protein. So let's look at another depiction of a sarcomere, and you can see here the Z lines, which in this depiction is called the Z disc. So we can call them either way, Z lines or Z discs. So here's the Z line on either side. You can see the wavy lines on the inside, which hold the thick filaments to the Z line. The lines, on the other hand, are attached to the Z lines. The thick filament begins here and ends right there. So this is the A band. The A band is the beginning and the end, from beginning to end, of the thick filament. The A band has two zones of overlap, one zone of overlap here, another zone of overlap on the other side. Zones of overlap are areas where thick and thin filaments alternate, thick, thin, thick, thin, thick, thin. In the middle, however, there are no thin filaments, there's just thick filaments. So the middle is called the H zone. So down here is an X-ray depiction of a sarcomere. And you can see the Z lines right here and right here. You can see the A band from beginning of the thick filament all the way to the other end of the thick filament. So we have two zones of overlap, one here and one here, flanking an H zone, which is an area of only thick filament. So you should definitely know the composition of the A band, what the H zone is in the middle of the A band also is important to remember. The I band looks at the thin filament only. So the I band begins at the beginning of the thin filament, ends at the end of a thin filament. So the I band encompasses two sarcomeres. Yet another depiction, so you need to count on seeing the picture of a sarcomere in the test and be able to identify the components. Go over the worksheets, make sure you can identify the parts of the sarcomere in the worksheet. So the Z line is right here, Z line is right here, so there's the beginning and the end of the sarcomere. The A band is right here in the middle in red. It has one zone of overlap, one H zone, and another zone of overlap. And the H zone, all it has is thick filaments. Now we need to talk in more detail about the composition of the thin filaments and the thick filaments. The thin filaments are made of a protein subunits called G-actin. G-actin are going to assemble into F-actin. The protein tropomyosin covers what we call the active sites of actin. So actin is this circular protein that assembles together into a filamentous shape. And on one side of the protein, there is what they call an active site. That active site is being blocked by a protein called tropomyosin. So tropomyosin is an elongated protein that sits on top of the active site and covers it. So that is a part of the structure of a thin filament. Now we have another component of a thin filament. This is another protein called troponin. Troponin is a protein that holds the other two components together. It's like a clamp, and the clamp clamps actin and tropomyosin together, creating this butterfly-shaped structure, which is helping to keep the tropomyosin in place, covering the active sites of actin. Let me show you the depiction of the thin filament. The yellow dots are going to be actin. The black dot in the middle of the yellow dot is the active site of actin. And notice that we have these structures in brown, which are going to be covering the active site of actin. And then the clamp, which is troponin, is sitting holding all the components together. Okay. So definitely know the components of the thin filament. Thick filament is much simpler. There's really only one protein in the thick filament, which is called myosin. And thick filaments are located so that the thick filament, that the head of the myosin protein is always adjacent to a thin filament. So the myosin is a protein that has a head here, and then what's called a tail, and the heads have a site in the middle which attracts or binds to the active site of actin. So in the zone of overlap, in the sarcomere, we're going to see thin filament, thick filament, thin filament. And as calcium becomes available, the heads of the myosin go up and bind to the active site of actin, creating a cross bridge which leads to a muscle contraction. So definitely know the parts of the thick filament and the thick filament, the names of the proteins that make thick and thin filament. Thin filament, the name of the protein is going to be actin. Thick filament, the name of the protein is myosin. Both actin and myosin have an active site that attract each other like the two poles of a magnet. Okay. The active site of the myosin, or the actin binding site of the myosin, also has an ATP binding site and an ATPase binding site. Okay, that's important to note in muscle contraction. Again, this depiction is showing you a thin filament. The circles are going to be G-actin. When G-actin assembles into a filament, we call it F-actin, the assembled G-actin. Tropomyosin is going around covering the active sites of actin, and tropomyosin is clamped together with the thick filament actin. Is all clamped together by troponin. Okay, so troponin has two hands. One hand grabs a hold of the tropomyosin, the other hand grabs a hold of the actin. The third hand is a calcium binding site. So troponin is going to bind calcium from when calcium is released from muscle contraction. So here's troponin, and here's the three heads of troponin. The C head in the middle is going to bind calcium, and when that happens, troponin moves out of the way, drags tropomyosin with it, and tropomyosin moves from its location covering the active site of actin, and now the active sites of actin are available. More of the same, just showing you the construction of a sarcomere, the thick and thin filaments. Down here in purple, the thick filaments, you can see the heads of the myosin towards the thin filament. This all becomes important in muscle contraction. All right. Let's see. Okay. So know the components of a sarcomere, know the components of the sarcomere that bind calcium, and the component of the sarcomere that binds calcium is the troponin. So right here in orange, that will be the troponin, which is going to bind calcium. When calcium binds the troponin, troponin moves out of the way, and as it moves out of the way, it drags tropomyosin with it, exposing the active sites of actin, which is what I had right here. And then the thick filament underneath, the head, will perceive the active site of actin close by, and it goes up and binds to the active site of actin. So all these structures are important in muscle contraction. And the theory that explains how skeletal muscle contracts is called the sliding filament theory of muscle contraction. And the sliding filament theory of muscle contraction has three parts. It has the events at the neuromuscular junction, it has excitation, contraction, coupling, which is in which calcium is involved, and then cross-bridge formation, in which again calcium is going to be available. Okay, so the first part of the sliding filament theory of muscle contraction are the events at the neuromuscular junction. The neuromuscular junction is the area where the nervous system reaches out and innervates a skeletal muscle, hence the name neuromuscular junction. Okay, and so here is the neuron that reaches out to the skeletal muscle cell and secretes a neurotransmitter that is going to stimulate the skeletal muscle cell. Again, this is a close-up of the neuromuscular junction. Here is the end of the axon or the axon terminal of the neuron that reaches to skeletal muscle. The neuron that reaches the skeletal muscle is called the somatic motor neuron. Okay, so here's the end of the somatic motor neuron reaching out and connecting with the skeletal muscle. So again, these are the events at the neuromuscular junction. Notice that when the signal goes through the neuron and reaches the end of the neuron, that causes an influx of ions, calcium ions, into the neuron. And when calcium ions reach the inside of the neuron, they're going to release a neurotransmitter. Okay. So again, this is a depiction of the neuromuscular junction. These right here will be the cell membrane of the muscle cell. This is the end axon of the somatic motor neuron, and the circles that you see inside are vesicles filled with neurotransmitter. A neurotransmitter is going to be a chemical that is going to be released and stimulate the muscle cell into reacting, into contracting. Okay, so there are several depictions of these events. So let's look at them in the pictures where we have these events going on one at a time. So the first thing that's going to happen, let me go back, is that a nervous impulse is going to come along all the way from the body of the neuron out into the axon, and it's going to travel all the way to, let's say, the lumbar region of the body. Okay. And at that point, the end of the neuron is going to be in contact or innervating with the skeletal muscle right there. So skeletal muscle and the somatic motor neuron, the end of the somatic motor neuron, are going to create a neuromuscular junction. So this is what you see here is a close-up depiction of the neuromuscular junction. This is the end of the neuron here. The vesicles you see inside are loaded with a neurotransmitter called acetylcholine. We call it ACH for short. So these little green dots will be the acetylcholine or ACH. Notice that a nervous impulse arrives at the end of the neuron, and that causes the influx into the neuron of calcium ions. So now we have an influx of calcium ions. Let me go back to the first picture. Okay, you can see here calcium going in as the nervous impulse arrives at the axon terminal. The release of the neurotransmitter can be seen in here. Okay. And the neurotransmitter is going to bind to the acetylcholine receptors on the cell membrane. So now these neurotransmitters bind to the acetylcholine receptors in the cell membrane. Now the receptors are going to open. So when the neurotransmitter binds to the receptors, these ion receptors open, and they're going to allow the influx of sodium and the outflux of potassium. That will be the end result of the arrival of the signal all the way to the end of the somatic motor neuron. Okay, so now we have the neurotransmitter bound to these ion channels, and when the neurotransmitter binds to the ion channels, the channels open, a tremendous amounts of sodium go into the cell. So much so that the cell is going to become very, very positively charged, and those charges will begin to travel through the cell membrane, and they're going to create more and more sodium gates to be open and more sodium going in. So that concludes the events at the neuromuscular junction. The second part of the sliding filament theory of muscle contraction is the step called excitation-contraction coupling. Okay, so we're going to look at the second part of the sliding filament theory of muscle contraction. So what happened was, in the previous event, the nervous impulse arrives at the end of the neuron, and as it does so at the end of the neuron, calcium goes in. That causes an influx of calcium into the neuron, and the influx of calcium makes the inside vesicles of neurotransmitter of the neuron are going to be released. So there are the little vesicles inside filled with neurotransmitter, which in response to the influx of calcium into the neuron is now going to release these neurotransmitter. And remember that the neurotransmitter is called acetylcholine, or ACH for short. These were the events at the neuromuscular junction. The neurotransmitter binds to a receptor in the muscle cell. The receptor opens and sodium goes inside the cell. Sodium begins to travel through the cell and begins to go down the T-tubules, causing depolarization of the membranes of the T-tubules. That depolarization is going to open calcium channels found in the cisternae, and that's going to cause calcium ions to escape into the interior of the cell from the storage area. So let me show you this right here. You see here the wave of depolarization, or the signal, nervous impulse, travels down the T-tubules. Here's a triad. So the events of the neuromuscular junction depend on a triad. As the nervous impulse travels inside, the nervous impulse is the sodium ions making the cell more positive. These gates, voltage gates, are going to open as the signal passes through close to them. They open, and the ions released are going to be calcium ions. So these red dots you see everywhere are calcium ions. So the excited contraction coupling is going to couple the nervous impulse to the release of calcium into the cytoplasm of the skeletal muscle cell. And that's what's called excitation contraction coupling. Okay. So now calcium has flooded the inside of the cell. It has been released from the sarcoplasmic reticulum. And now the last step of this sliding filament theory of muscle contraction is the cross-bridge formation. Okay, so now you have calcium flooding the inside of the cell. Remember that troponin is the protein in thin filament that binds to calcium. So calcium binds to troponin, and now troponin moves out of the way. So here troponin has moved out of the way and has released the binding sites of actin. Okay. So the active sites of actin are now being exposed, they've become available, and remember that the head of the myosin protein has an active site of actin receptor. So when this active site of actin is available, like two poles of a magnet, these two structures come together into what's called a cross bridge. So you can see here the head of the myosin going up and binding to the active sites of actin. So the actin and myosin coming together is what we call the cross bridge. So this is the formation of the cross bridge. So cross bridge formation is the last part of the cycle. Cross bridge formation involves the release of the calcium, the binding of the calcium to troponin. Troponin then moves away, and the active site of actin is available for the myosin head to attach. What happens next once the myosin binds to the actin is that the myosin head, here's the binding right here, myosin head is going to move forward towards the H band, and that's going to create a release of end products from the myosin head, and now the myosin head is available to receive ATP. So ATP binds to the myosin head, and the myosin head moves forward and then detaches and goes to rest. So let me show you here real quick. Okay, so what has happened here? Calcium bound to troponin. You should definitely know what each of these proteins do. So troponin binds calcium. Okay, when that happens, the active site, troponin binds calcium. Right here, when that happens, troponin moves out of the way and dragging with it tropomyosin. That's the little rope in tan color in the picture. When tropomyosin moves out of the way, now the active sites of actin are available. A myosin head comes along and attaches to the active sites of actin, creating a cross bridge. Next thing that happens, the myosin head releases two molecules that are attached to itself, ADP and phosphorus. And then the molecule detaches and moves, and as it does that, it pulls the thin filament towards the middle. So sarcomeres are contracting towards the middle of the sarcomere. So this is again the depiction of how all of this is happening, and I'll let your dad take a look at that. You can take a look at that through these pictures. You go here, right there. Okay, so this is showing you the entire cross-bridge cycle. Again, I cannot emphasize enough, you should know which protein binds calcium, which protein of the thin filament binds calcium, and that's going to be troponin. You should know the role of tropomyosin, which is to cover the active sites of actin, and when troponin binds to calcium, it drags tropomyosin out of the way. Now the active sites of actin are being exposed, and that makes a cross bridge. You should definitely know which two proteins make the cross bridge, which will be the actin molecule and the head of the myosin. Okay. When the actin when the cross bridges are formed, go over here, and the myosin head moves forward, that's going to pull the thin filaments towards the middle. So take a look at these. This is a sarcomere before contraction. You can see the A band right here. You can see the zones of overlap on either side of the H zone in the middle. During muscle contraction, the myosin heads make cross bridges with the thin filaments, and then the myosin heads tilt and pull the thin filaments towards the H zone. What that does is several things. First of all, the H zone becomes smaller as the thin filaments are pulled towards the H zone. Then the Z lines come closer to each other. So Z lines come closer, okay? And the I band becomes smaller. Here's an I band, here's the other I band. Compare that to this I band right there. Okay, so I bands become smaller. Put it down here. Okay, as the entire sarcomere is pulled towards the H zone, and so you can see here several sarcomeres, and as each sarcomere is being pulled towards the H zone, the entire myofilament contracts towards the center. The A band does not change because the A band, by definition, is the area between the beginning of the thick filament to the end of the thick filament, and the thin filament is not changing size during contraction. So you should know what happens to the sarcomere during muscle contraction. The H zone becomes smaller, Z lines come closer to each other, and the I bands become smaller. The A band, on the other hand, does not change in size. Okay, so you should definitely know the events that happened during muscle contraction, and definitely know here the three parts of it. The events at the neuromuscular junction, the excitation, contraction, coupling, and the cross-bridge formation. And given a description of the event, you should match it with its name. Okay. So this is the first part of the PowerPoint. Let me, right here. Let me point something out real quick. This is these three events at the neuromuscular junction. Notice these sites right here and right here. These represent animations of each of these events. It's not a bad idea to copy and paste these websites and look at the animation, which further explains how these events happened. So in the test, you're not going to be asked to explain these events, but you are going to be asked questions like, what's the role of calcium in muscle contraction, for example? It binds to troponin. It allows for the, prior to that, in the events at the neuromuscular junction, calcium releases the neurotransmitter, ACH, acetylcholine, which is the neurotransmitter. Okay. Calcium ion, this happens outside. This happens at the outside of the muscle cell and the synapses. Okay, between the neuron and the muscle cell. Inside the muscle cell, calcium is going to be released from the sarcoplasmic reticulum and is going to initiate muscle contraction. So calcium is going to be needed inside the muscle cell to initiate muscle contraction. And as long as calcium is present, muscle contraction will continue. Okay, so that's the role of calcium in muscle contraction. Now, another question would be, or could be, what is the role of ATP, energy, in muscle contraction? Okay, so let's go back to how muscle contraction happened. Let me go back to the cross-bridge formation. And actually what I want to do is I want to go to the, there we go, this right here. Okay, so here you have the calcium that is binding to the troponin and has moved the tropomyosin out of the way, and the active sites of actin are available, and the cross-bridge has formed. So far, no ATP has appeared. So no energy has been used to create the cross-bridge. Okay, next thing that happens is that the cross-bridge moves, the head of the myosin moves towards the middle, creating the muscle contraction. Notice no ATP is needed for this step either. In this step, however, here's ATP needed. So ATP is needed to release the cross-bridge. Because if you think about it, the cross-bridge is being made by two molecules that attract each other, like two poles of a magnet. So these right here are attracting to each other, like if they were magnets. No energy is needed for that. The energy is going to be needed to release those two molecules that are attracting each other, to release them from each other. That's what ATP is going to do. So ATP breaks the cross-bridge. Okay. And that's important because what that means, for example, is that if ATP is not available, the muscle will remain contracted. It will not relax. ATP is needed for relaxation, or the muscle to break the cross bridges is not needed to make the cross bridges. Upon death, a body becomes stiff because ATP is not available anymore. So all the cross bridges have remained intact without relaxing because there's no ATP to relax them, and that's why all the muscles end up contracted. That's called rigor mortis. Okay, so that's important, the role of ATP in muscle contraction. There is another role of ATP in muscle contraction, which is going to be explained on the next set of PowerPoints, but I'm going to go ahead and address it here, right here. Okay, calcium is released from the sarcoplasmic reticulum into the inside of the cell. It is stored inside the sarcoplasmic reticulum, and it's going to be released into the cytoplasm of the cell. The reason why calcium leaves is by diffusion, because there is lots of calcium inside the sarcoplasmic reticulum, less calcium in the cytoplasm. So by diffusion, calcium will go from an area of high concentration to an area of low concentration. After muscle contraction, all that calcium has to be put back into storage, and then we have to go against a concentration gradient. That's going to cost ATP. So ATP will be needed to return calcium to the storage site. So you should know why calcium is needed during muscle contraction. And again, it's needed for two purposes: to make the acetylcholine to be released from the neuron, and to release the active cytobactin inside the muscle cell. You should know the role of ATP in muscle contraction. And here is again ATP being needed in order to release the cross bridge. Okay, so again, these are complicated events, but I think it helps if you were to look at the animations that are provided. Are there any questions? Okay, I'm going to move on to PowerPoint B and address at least some of PowerPoint B slides. We may not finish it. We're nearing about an hour, and I don't want to go past an hour, but we can at least cover some of these PowerPoints, and then we can have another session next week, maybe on Monday of next week, and finish up this chapter. Okay, so this next PowerPoint talks about some of the mechanics of muscle contraction. First thing that it talks about is a concept called motor unit. Motor unit consists of the somatic motor neuron that serves a muscle cell, and all the muscle cells that motor neuron innervates. Okay, so let me go to the picture first. And so here we have two neurons, somatic motor neurons. We have the red one, and the red one is innervating 1, 2, and 3 muscle cells. So that is one motor unit, which is a 3-cell motor unit. Here we have the purple one, and the purple one is innervating 1 and 2 muscle cells that I can see. Okay, so the purple one, the second motor unit, is innervating 2 muscle cells. So those are 2 motor units on this muscle here. Okay, motor units can serve as many as 4 to several hundred muscle cells. So again, you have the neuron, somatic motor neuron, and all of the muscle cells that neuron innervates. That's a motor unit. Some motor units are large because the somatic motor neuron is in charge of hundreds of muscle cells. So you have one neuron innervating 100 muscle cells. A small motor unit is one in which the neuron is only in charge of stimulating a few muscle cells, let's say 5. Okay. Muscles that require a lot of precise movement will have small motor units. Look, so muscles of the finger or muscles of the eyeball, extraocular eye muscles, will have small motor units. Muscles that do not require fine motor movement, but rather are large weight-bearing muscles, are going to have large motor units. For example, the muscles of the thigh or the hips or the calf. So you should definitely know the principle of what a motor unit is and the difference between a large motor unit, a small motor unit, and the types of muscles where you find large or small motor units. Part of the role of a motor unit is to prevent muscle fatigue. So motor units are going to contract in an asynchronous manner. They're going to take turns contracting, and that will prevent muscle fatigue. So here in this depiction, you have this red motor unit, number 1, contracting first, then goes into relaxation, and the blue motor unit cells begin to contract. Then as it goes into relaxation, the purple motor unit cells begin to contract, and then back to the first motor unit. Okay, so this is a synchronous contraction, which prevents muscle fatigue, because these motor units can take turn contracting and then have a time to rest before another contraction. This helps to sustain contractions for a long period of time without fatiguing the muscle. Okay, another important concept is that of a twitch. A twitch is going to be the contraction and relaxation of an entire muscle. So this is not a muscle cell, this is the entire muscle. Okay. So in order for the muscle to contract, there has to be a stimulus. So these will be the events at the muscular junction. Acetylcholine is released. This muscle cell is depolarized. The depolarization, the stimulus travels down the T-tubule, and all these takes time. So this is going to be called the lag period, where no contraction is seen. Following the lag period, a contraction will be seen when we are into the formation of cross bridges, and then contraction peaks and the muscle relaxes again. So that is a twitch. The part of a twitch is the latent period, which is the time when the events at the excitation contraction coupling are taking place. Then we have the period of contraction, which is cross bridge formation and the production of the actual tension. And then the period of relaxation, where calcium is going to be moved into the sarcoplasmic reticulum and the tension begins to decrease. Definitely be familiar with the depiction of a twitch. So you should recognize the graph as that of a twitch. Here's the stimulus, the little latent period. Then we have the period of contraction, then the period of relaxation. Okay. Twitches can last a long time or be very, very quick. So they can be of varied lengths. So we have a small twitch, medium-sized length twitch, and a long time, a twitch that takes a long time. In general, muscles for which have very good muscle control, very accurate muscle control, have small twitches. So the extraocular eye muscles, the lateral rectus, superior rectus, inferior rectus, medial rectus, superior oblique, inferior oblique, all of these muscles that move the eyeball have very small twitches. Muscles that bear weight, like the calf muscles, the gastrocnemius and the soleus, are going to have longer twitches. So you should know examples of muscles with long twitches versus examples of muscles with small twitches. Okay. Now, the next thing that happens is that scientists begin to play around with the frequency of the stimulation and the strength of the stimulus to see what happens to the twitch, okay, to the muscle contraction. So a single stimulus results in a single contraction response, which is a twitch. Now, what happens if the stimulation changes, becomes a stronger stimulus? So we're going to change the strength of the stimulus to see what happens. What if the frequency of the stimulation changes? So let's look at what happens with the first event when we change the stimulus of the frequency, the frequency of the stimulation. So in this example, stimulation happens. There's a lag phase, there's contraction, muscle begins to relax, And before the muscle completely relaxes, another stimulus is generated, which means more acetylcholine being released, which leads to more calcium being released in the cell. And notice what happens to the next contraction. It becomes stronger. Okay. So that leads to stronger contraction. Then before that second relaxation period is finished, another stimulation happens, leading with a next contraction being stronger than the previous one. We do the same thing again before the muscle relaxes, another stimulation, and this one is even stronger than the first one. So what you can see is as we increase the frequency of a stimulation and we don't allow the muscle to completely relax, the subsequent contractions become stronger and stronger and stronger. The reason why is because calcium has been released during each contraction. During the relaxation period, the calcium is supposed to be put back into storage and disappeared. However, instead of that happening, another stimulus comes about and more calcium is released into the cytoplasm of the cell, causing a stronger contraction. The more calcium, the stronger the contraction. Okay? So this continues to happen, creating a progressively stronger and stronger contraction until the peak of tension that muscle can produce is reached. This is called incomplete tetany, and that is important. Incomplete tetany is the name of this event. Now let's play with something else. Let's do something different. Now let's increase the frequency of stimulation so much that the cell doesn't even begin to relax. So what happens is there is a stimulation, there is a lag period, there is a contraction, and then all of a sudden another stimulus which releases more calcium. The cell continues to increase its contraction as more and more stimulus arrive and short duration. The cell quickly reaches maximum tension and retains that maximum tension for as long as this quick stimulation event happens. This is called fused or complete tetany. So you should be able to distinguish between incomplete tetany, here, in which partial relaxation takes place, versus complete tetany, in which no relaxation takes place. We do this to our muscles all the time. So for example, you're trying to lift a heavy object. So you created a stimulus and the tension created is not enough to lift the object. So without relaxing, you stimulate again, creating a stronger contraction. That doesn't do it. You stimulate again until you reach peak contraction, and that may allow you to lift the object. Now let's say you're lifting the object and you want to keep it lifted. You want to lift it above your head and keep it above your head. Now you need to create complete tetany. You need to stimulate your muscles in such high frequency that the contraction happens and the contraction remains at its peak and sustained over a period of time. So that's complete tetany. So definitely know about these events and how they are created, and be able to identify the graph and match the graph to complete tetany versus incomplete tetany. Okay. Let me go to, let me finish this portion and then we'll stop there. Okay, so the response to changes in stimulus strength. Okay, before we change the frequency of the stimulation, now we're going to change the strength of the stimulus. Okay, so this line, white line here, represents what's called the threshold stimulus. This is the minimum force of stimulation needed to create a contraction. So we send a stimulation out, and that's not enough to cross the threshold, so no contraction happens. So we increase the stimulation, and notice how here none of the motor units in the muscle are being excited because the stimulus is not strong enough. The third stimulus is strong enough to reach the threshold, and now you have two muscle cells contracting. You need more force than that, so you continue increasing the force of stimulation, and as you do that, more and more motor units are recruited into contraction. So what that means is that as we create a stronger and stronger stimulus, more and more motor units are going to be recruited into contracting. okay, creating more and more strength of contraction. And again, we do these all the time when we're trying to lift something heavy. So the size of principal motor units, okay, with as we continue to stimulate the muscle, larger and larger fibers are recruited. So first stimulus gives you motor units with the small muscle fibers, meaning small muscle cells. So within a muscle, you have different sizes of muscle cells. And the first ones to be recruited, stimulated, are going to be the small ones. Okay, well, that doesn't do it. That doesn't lift the object you want to lift. So you increase the strength of stimulation, and that's going to recruit medium-sized motor units. And let's say that doesn't accomplish the work you're trying to accomplish, so you increase the stimulation, the strength of the stimulus even more, and that recruits the even larger motor units. And then you can finally do the job you need to do. Okay, so you need to be familiar with these concepts and identify them when they're explained in a test, for example. Any questions about these? Okay, so I'm going to go ahead and stop the recording now, and I will return and have another session.